Introducing of the land plant rooting system. Welcome.
Thanks very much, Merle, and thank you all very much for coming. I'm not going to use the microphone, so if you can't hear me, make a sound and I'll put the microphone on. So, around 470 million years ago, one of the pivotal events in Earth history occurred, and that was the invasion of the land by plants. Before that time, all primary productivity took place in the ocean, and since that time, 50% of the productivity has taken place in the terrestrial realm, and it's land plants that are behind all of that carbon fixation. The transition to land involved a number of innovations, and amongst those innovations were the evolution of tissue systems, meristems with stem cells that gave rise to those tissue systems, structures for increasing surface area to absorb light for photosynthesis, and what I'm going to talk about today are rooting systems. So these pioneer colonizers anchored themselves and absorbed nutrients through a variety of structures, and I'm going to call them rooting systems in general. And if we look at the fossil record, the earliest rooting systems that we see were really quite simple. So what we're looking at here is a piece of fossilized soil that's fixed in chert. So this is basically silica. Here we've got the bottom of a rhinophyte plant, and they produce these little knobs, and on these knobs there are these structures, these filamentous cells, these rhizoids. So the earliest rooting structures that we know of look like this. So this is taken from the Rhine chert, which is an immaculately preserved ecosystem from around 409 million years ago. So that's the early Devonian. The next level of complexity that we come across is the formation of true roots as we know them today. And we can reconstruct these roots by examining coal balls. So coal balls, here's a slide through a coal ball. So these are nodules of calcium carbonate that formed in the peaty soils of the Carboniferous coal swamps. So Carboniferous coal swamps formed not very far from here, down south and towards Pennsylvania, up through Europe as far as Poland. And these coal swamps had a very diverse flora, and these diverse floras, elements of these floras were preserved in situ in these small coal balls. So what we have here is a piece of fossilized peaty soil, and all the plant material in there is preserved. And you can make thin sections or grind down thin sections, and what you're seeing here are cross sections of root axes. And if we use these, look at these images, we can see there's huge amounts of information. So for example, earlier this year we described the earliest fossil root hairs from one such structure. So these are, this is the epidermis of the Stigmarian root system. This is our reconstruction of that root system. So they form these tall palm-like trees and then develop these highly bifurcating branching systems, seen here, flowing up from here. And then on the surface of these structures, you've got root hairs forming here. And it's in these coal balls also that we see the oldest preserved root meristem. So this is the oldest root meristem that's been preserved to date. The organization of cells in this root meristem are different to any extant plant. It's likely to be a pteridosperm, so this is an early seed plant, but we're not sure. But the take-home message here is that by 380 million years ago, before these roots were formed, roots, specialized axes with meristems and stem cells at the tip that were covered in filamentous cells had evolved. And if we now look at extant plants, so plants that are growing today, we see the same general pattern. So this is a simplistic representation of the phylogeny of land plants. So we've got the earlier divergent groups here, the bryophytes. Then these groups here are the vascular plants, the lycophytes, ferns, vorstales, and then the seed plants. And the first three branches of the land plant tree only form these filamentous rooting systems seen here as rhizoids and marcantia. These are multicellular rhizoids in the moss Fiscomatrella. And then in the seed plants, here's Arabidopsis and rice. We see we've got these specialized rooting axes, and on the surface of those rooting axes are those elongated cells. So for those of you who want to go to sleep for the next 45 minutes, I'll tell you what the aim of my talk is and the aim of our research is, and I'll wake you up at the end. So our aim is to begin to reconstruct the genetic system that controlled the formation of these early ancestral rooting systems. And the approach will be to understand the mechanism in plants down here, understand the mechanisms in plants down here. What's common between these two will have been inherited from the common ancestor. So, the first part of the talk is going to talk about our recent work on characterizing the genetics and the formation of these systems in Mercantia. So, Mercantia is a beautiful system. It's haploid, like all of the bryophytes. Here's our wild-type plants. So these form small, flat leaf-like structures. They've got meristems in their notches. Okay, and then on the underside, they form this masses of rhizoids. And you look at any rhizoids forming on the dorsal surface. So they're only forming on the ventral surface. So then we can make mutations, and you can use any mutagen you want. You use CMS, UV, tDNA, and you get mutants that look like this. So the difference between this and this is pretty obvious. There are no rhizoids here, and there are lots of rhizoids here. Okay. So to do this screen, and the results I'm going to talk about today are derived from this screen. So we generated 336,000 tDNA transform lines, and then identified 329 defective rhizoid mutants from these. But in order to identify the genes that we mutated, we needed to put together a genome sequence. So we just went ahead and sequenced the genome. And the important data here is that their N50 is around 366 kilobases. So most 50% of the contigs in this are greater than 366 kb. So this means that we've got a pretty good chance of spotting everything that we need. Okay, so we made ESCs and used them to annotate the genome. So we had a genome that we could use. Okay, so here's the sorts of information that we've got. So if you look down here, we've got the various classes of mutations that we found. So we've got very few rhizoidal mutations, so out of over 300 mutants, we only found 6 rhizoidals. 3 of these we were able to cross and show that the phenotype was co-segregated with the hygromycin resistance gene on the tDNA, so they were likely tagged. 3 we didn't cross, but a total of 6. We found some mutants that had extra rhizoids, okay, and then we got lots of mutants that have defective rhizoid shapes, so the short rhizoids, branch rhizoids, bumpy rhizoids. Okay, and for most of the talk today, I'm going to focus on these top 2. So the first mutants that we identified with like this, I've shown you this one before. The main difference is that there's no rhizoids here. And when we sequenced the insertions, we found that the insertion sites, the tDNAs had inserted into an RSL gene. This is a basic loop helix transcription factor. So we both alleles inserted into this basic helix transcription factor encoding gene. So we figured, okay, 2 independent alleles, it's likely to be the right gene. Just to make sure, we took the coding sequence and overexpressed it using a rice-actin promoter, introduced it into a mutant, and you restore the formation of rhizoids. So we'd identified the right mutant, so that was fine. So I was really very excited by the fact that we found this group of mutants here. So we found 20 mutants, 18 of which were crossed and co-segregated. So I was a bit disheartened and disappointed when I saw that these 2 were insertions into the vicinity of the RSL gene, but this time these insertions were into the 5' region up here. So we've got 2 insertions into the 5' non-translated region, and then we had 3 of the characterized mutants that were into this 5' region here. And based on their location, we predicted that these were likely to be gain-of-function mutations. So, and of course they are. So what you've got here is the expression levels of RSL1. Here's our wild type, so there's boys and girls here. So they've got relatively low levels of expression. And then in each of these mutant lines that produce this abundance of rhizoids all over the shop, these guys are all gain of function. So normally the mutant won't ever make a rhizoid on its dorsal surface, but when you express large amounts of the RSL transcription factor ectopically from the 35S promoter, you get this phenotype. So we were pretty sure that this was a transcription factor component of the phenotype. So what we can say now is that RSL1 is positively regulating the development of rhizoids in Mercantia. So the question was, what's it doing in other plants? Well, we previously knocked out exactly this gene in Fuscomatrella. So here's a family tree of these. transcription factors. So the RSL gene belongs to a group of genes called the class 1 RSL genes, RSL1 class 1. And there's a single gene in Mercantia. There are a pair of genes in Fiscomatrella, and if you knock out both of those genes, you get plants that look normal, except for the fact that they produce few and quite short rhizoids. So this is consistent with the hypothesis that these transcription factors positively regulate rhizoid development. In addition, if you overexpress these genes in Mercantia, or sorry, in Fiscomatrella, you transform plants into bundles of rhizoids. So this is what Fiscomatrella plant looks like. If you drop a spore onto agar medium, come back 20 days later, you get something that looks like bread mold. And there are two types of filaments here. There are filaments that move radially from the centre to the outside, and so here's a close-up of one of those. And they produce buds, and these buds will then develop into the plants with leaves. Okay? So that's wild-type development. In the plants that overexpress both RSL1 and RSL2, we transform these buds into masses of rhizoids. So every cell here produces a rhizoid.
When you're looking for Mercantia mutants, did you find any rhizoids with right angle crossbows?
With right angle? No. The rhizoids in Mercantia are unicellular, so they're not multicellular like.
Right. But they have crossbows.
Not in Mercantia. In the Fiscomatrella they do. Okay, yeah. But that's an interesting question with respect to these. And so if we overexpress, if we look at the spores, if we look at early stages in the development of these, in Mercantia, the loss of function in Fiscomatrella, we only ever get transverse walls, we never get oblique walls. So in Fiscomatrella, the chloranema cells produce transverse walls, the cholanema cells produce oblique walls, and we only ever get the transverse walls in that case. So it does affect oblique versus transverse orientation, that's your question suggested. So then if we take either of the Mercantia or the Fiscomatrella genes and then introduce it into Arabidopsis, we can restore the development of root hairs in mutants that otherwise don't make root hairs. So here are the two Arabidopsis genes, RC6, RSL1, that are most closely related to RSL1 from Mercantia. So here's our wild-type Arabidopsis. Here's a plant that lacks both of these genes, and now if we introduce the Arabidopsis gene back in off a 35S promoter, we can get root hairs forming, and if we introduce the Mercantia gene, we get root hairs forming and very indistinguishable in phenotype. So what this tells us is that the function of these proteins has been conserved over the approximately 450 million years since these two plants last shared a common ancestor. I haven't gone into it, but these proteins are conserved in their basic helix-leucohelix domain. There's no conservation outside of those domains. So the function has been conserved for some time. So what I can say now is that given that we've shown that the RSL genes are controlling the formation of these rhizoid structures here and here, we can say that the RSL genes likely control the development of these filamentous rooting cells in the common ancestor of the life. So that was all fine and dandy. What else do we have in our mutant collection? Well, we have these mutants here. So these were rhizoidous mutants that were crossed and co-segregated. So we wanted to look at some of these. These are gemmi. Gemmi are asexual propagules that are produced by Mercantia. And so this is why Mercantia is such a weed. So they grow in your greenhouse and your gardens, and water falls on them and they just spray everywhere. So it's a bigger weed than Arabidopsis. It's really quite a challenge. And so this is a close-up of the propagules here. And if you look closely at these propagules, you can see that there are these pale cells here and a background of darker green cells. And so these pale green cells are feature rhizoids. So this is 5 days later. So these are rhizoid precursor cells, and then these are the rhizoids a few days later. We found that there were 3 of these mutants. These were allelic. Okay, they didn't form rhizoids. That's how we identified them. And if you go back to this stage, we don't see any precursors. So they don't form precursors. They don't form any rhizoids. Okay, and they're allelic. And we identified the insertions of these genes. We found 4 genes, 4 nucleotides were caused by insertions, tDNA insertions, upstream of an open reading frame that didn't look like anything we knew. So given that I'd previously shown that the tDNA insertions in the 3' region of the RSL gene resulted in an elevated expression in RSL, we hypothesized that these guys are going to be gain of functions, and it's true. So if we look at the expression levels, and here, this is the wild type, boys and girls, very low levels, and in each of these mutants, we've got high levels of expression of this 1.2 kb transcript here that doesn't look like anything. Okay, so we thought, okay, this is good. So we're getting a phenotype that's correlated with the increased expression in a 1.2 kb transcript that doesn't look like anything that we can recognize. So just to make sure that we actually had gain of function mutations, we introduced the 1.2 kb transcript into Mercantia under the constitutive actin promoter. So here's wild type. You've got those precursor cells and you've got your rhizoids. Now we're taking the 1.2 kb transcript and we're ectopically expressing it at high levels. And you can see that we're repressing the formation of the precursors and of course no rhizoids form. Okay, and so just to show you that these really are overexpression. So here's our wild type controls, relatively low levels of expression. And if we look at transform lines that have very few rhizoids, we can see that they're all overexpressing the 1.2 kb transcript, whereas transformants where we get a wild type phenotype, we can't detect any overexpression. So our model that these mutants were gain of functions, I think, holds up. So the question is, what did this look like? So we had this genome sequence, we went through all that sequence, all that trouble, and when we identify the second gene we identify, it doesn't look like anything. We decided we needed to break it up into little pieces and to see what would work. So what we did then is, the experiment I just showed you was where we took the 1.2 kb transcript and then introduced it using the actin promoter, okay, and we repressed rhizoids. So the code I'm going to use is, in green there's no rhizoids developed when using a 35S or an actin promoter. Okay, so then we took that transcript, we split it into two, and there was a right 0.7 kb transcript and a left 0.7 kb transcript, and overexpression of this transcript repressed the formation of rhizoids. And then we split this up into a few fragments, and we were calling them ORFs because at this stage we didn't know whether they were open reading frames or not. And so we could split up this into 3 fragments, and then one of these fragments, when overexpressed, repressed the formation of rhizoids. And so here's the data for that. So you've seen wild type here. So here's a wild type with the precursor cells and the rhizoids. Here's the plants that are overexpressing the entire 1.2 kb region, and it represses the rhizoid development. This is this ORF1, this small fragment that I've shown you. And if you take that, overexpress it, you again repress the formation of rhizoids, indicating that there's activity, rhizoid repressive activity in this small fragment. And then if you take either of the other 2 fragments, you don't have any impact on rhizoid development. So then we could figure that actually this small region had some rhizoid repressive activity. So given that it was a small region, we figured it might have been a microRNA. But none of the microRNA hadn't appeared in any of the libraries that people had been making. So we contacted them and said, look, we suspect that there might be a microRNA in here. Can you have a look? And of course there was, but they were finding it in such low abundance. So we're finding 5 reads in some libraries, 1 read, 7. So it was below their threshold. So that's why they missed it. So what we find is here's our full 1.2 kb transcript, and then there's a small region in here that corresponds to the FRI1 ORF that I talked about, and it produces a microRNA. Okay, so we were happy enough with that. So based on their high throughput sequencing, they were seeing a microRNA, a putative microRNA that looked like it was encoded in our 1.2 kb transcript. So we just wanted to convince ourselves that it was there, so we did some stem loop PCR, and we can detect the microRNA. So here's wild type boys and girls in the first 2 lanes, and then we can see the expression levels. of the microRNA in the gain of function lines. So it looks like it's a microRNA and it's expressed. So then when we looked at the sequence and tried to identify what its target might be, then it all began to make sense. So here's our microRNA sequence here, and then it predicts a putative target. And of course, this putative target sits right in the basic helix, in the basic domain of the RSL gene. So what we've got here is putatively a target. In our genetic screen, we've identified a positive regulator of rhizome development, and we've identified the negative regulator of that positive regulator. So if RSL really is the target of microRNA for H1, then we would predict that the RSL1 microRNA should be cleaved in this target site. And of course, by degradable sequencing, we find exactly a cleavage site corresponding to the predicted region in which we would expect to find them. So this is suggesting that we've got a positive regulator and a negative regulator. So if the microRNA is negatively regulating RSL1, then we might expect to see some effect on RSL1 transcription in the FRH1 gain of function mutant background, and that's what's presented here. So we're looking at the expression levels of RSL1. So here's wild types here. Okay, and if we look at the expression levels of RSL1 in these various gain of function, these are the 4 gain of function mutants that we found, and then these are the overexpressor lines, the transgenic lines that we made. We can see that the levels of RSL4 transcription are down by about 75%. So they're quite dramatically reduced here. So that's consistent with the hypothesis that RSL1 message is being cleaved by the FRH1 microRNA. And then we decided to do the opposite experiments to determine the effect of RSL1 dose on FRH1 expression. So here's our wild types here. Okay, and we find that in the RSL1 and RSL2 complete loss of function alleles, we get a decrease in FRH1 steady state RNA levels compared to wild type, and in the RSL1 gain of function mutants, we've got an elevation in the steady state levels of FRH1 RNA. So this suggests that RSL1 is actually positively regulating the transcription of the negative regulator, its own negative regulator. So we've shown you that RSL1 positively regulates FRH1, and that FRH1 is negatively regulating RSL1. So we've got ourselves a feedback loop here. So this is more or less where we are with this story, and what we're trying to do now is to explore the biology of this a bit more. We've got some preliminary data that's consistent with this model. So if we look at a FRH1 gain of function plant, it's rhizoidal, and now if we overexpress RSL1 in that background, we can get some rhizoids developing. So it's consistent with the idea that if you upregulate this experimentally, then you can basically outmaneuver the negative regulation from FRH1. So I was very excited that we found a negative regulator of RSL1, because having worked on RSLs and Viscotrella and in Arabidopsis, we'd never identified a microRNA that regulated those. So we looked to see was there FRH1 microRNA in other organisms, and there are, but they're all liverworts. So the FRH1 microRNA is restricted to liverworts, so we see it in the 2 major groups of liverworts, the complex haloid liverworts and then the leafy liverworts. And the FRH1 microRNA target sites is entirely conserved in these mutants, and you see outside of this conservation box, you don't see that much conservation. So, and we think it's unlikely that the Arabidopsis RSL1 gene is controlled by a microRNA, although we're not 100% sure. So that was the geneticists in me was all very excited. We found a positive regulator, we found a negative regulator, we got loads of alleles, and that was all very nice. And we thought we knew what it was doing. These genes are controlling the formation of these rooting structures. Great. Okay, and so then we made the fatal mistake of doing another experiment. You should quit while you're ahead. So this is a rather trivial experiment where we were looking to see what cells was the FRH1 promoter active in. And you can see these nice spots. So this is a close-up of the surface of those gemmy, so those green discs that I described earlier. And these are those large pale cells. So that's a rhizoid precursor. And then these cells in here are the epidermal cells. These are equivalent to the pavement cells of Arabidopsis. And so we can see that the promoter is active in these cells, which suggests that the role of FRH1 is to negatively regulate the role of FRH1 microRNA is to negatively regulate RSL1 in the rhizoid cell itself. But then we noticed that there was what looked like specific expression in other structures. So here is the crotch or the notch that you see in these discs. And I said earlier, this is where the meristem forms. And right next to the meristem, we can see there are these two structures. These are tri- or quadricellular papillae. So they're like little trichomes that secrete mucilage in and around the apical meristem. And these cells, at different stages of development, will express FRH1. So up until now, this had all made sense. We were seeing FRH1, RSL1 active in these future hair cells, future rhizoids. And now we were having to change the story. These are expressed in these other structures, and these are multicellular structures. So this is a close-up here. So if you now look at wild type, so these are these two structures that I told you, and showed you that the FRH1 promoter was active in those. If you now look at the FRH1 loss of function mutant, you see that this notch is clear. So that these mucilage papillae don't form in the FRH1 mutant. So that told us that actually the FRH1 gain of function mutants is repressing the formation of these bicellular structures. So then we got even more worried, and we thought we should go back and look at RSL mutants. And of course, we're seeing the same effect. So on top, you've got your wild type. This is, you can see 1, 2, 3 cells here in this mucilage structure. If you look at the same region of an RSL1 loss of function mutant, you don't see anything. So these aren't developing. And then things just went from bad to worse. We found in wild type, so there are these gemma cups. So these are the structures in which the gemmy form, those asexual propagules form. And so in wild type, these are chock full of these things. Okay? And if you take a slice through them, you can see them inside here, all packed in close. These are the walls of the cup here, and then these are the gemmy. And these gemmy develop as outgrowths from single epidermal cells. So single epidermal cells will swell out of the plane of the epidermis and then undergo cell division, forming these multicellular structures. In the mutants, in severe cases, you don't get any of these rhizoids, or these gemmy. So we'd always known that RSL mutants tended to produce fewer of these gemmy than normal, but we thought nothing of it. But then, having seen the papillae disappear, we thought we should look at these in more detail. And if you take a slice through this, see there's nothing in here. Well, this is chock full of these densely packed gemmy. There's nothing in here. And if we now look at the cells along this structure here, these are epidermal. They look like normal epidermal cells, and rather unlike these epidermal cells that are swelling out to form these structures. So this led us to conclude that the RSL genes and the FRH1 microRNA are not rhizoid specific, but actually what they're controlling is the formation of outgrowths from specialized epidermal cells. And then those outgrowths can either grow in the absence of cell division and form rhizoids, or they can grow in the presence of cell division and form these multicellular structures such as papillae and the gemmy. And so then we thought, well, actually, maybe we should go back and have another look at what's going on in the moss. And so we did that. And so I've shown you this before. So here's the double knockout mutant, and it forms these short rhizoids. But it turns out that Mercantia produces bicellular cells, hairs, like little papillae in the axils of these leaves. And, they're hard to see. So here's one cell here, and then here's another cell here. Okay, and you see them occasionally in the mutants, but the numbers, there's about a third of the number in the double mutant than there is compared to wild type, and this is pretty reproducible. So we missed this phenotype first time around. So this suggests to us that actually the RSL genes are controlling the formation of specialized epidermal structures. And that's schematically represented here. So what I've shown you is that these genes are controlling the differentiation of rhizoids, gemmy, multicellular rhizoids here, these multicellular hairs here, and the root hairs here. And what's common among all these structures is that the process by which a single epidermal cell bulges out and forms these structures. Now you might say, well, okay, that's fine, but these are really weird and obscure structures. But actually amongst the bryophytes and amongst the pteridophytes, huge numbers of structures are derived from single epidermal cells. So for example, the sporangia in eusporangia ferns are entirely derived from a single epidermal cell. The male and female reproductive structures in all the bryophytes are derived from single epidermal cells. So there's huge numbers, there's huge amounts of structures that are derived from these cells. And so one of the challenges for us now is to begin to understand the role, once RSL has kicked off this process, what's the mechanism by which the RSL gene promotes the formation of this polar outgrowth from this outer face. And then once it's done that, how does it decide that the outgrowth is going to be a unicellular structure or a multicellular structure? So we know squat about this process, and we're going to use some genetic analysis using specific suppressors of the gain-of-function mutants to identify genes that act in this pathway here. But we already know something about what's happening in this structure here. And I'm going to just give you a little taste of what we've discovered so far. There's one recent story that I think is quite nice cell biology associated with it. Okay, so a number of years ago we were interested in identifying genes that acted downstream of these, what we call class 1 RSL genes in Arabidopsis. Early on I told you that you RSL1 and RHD6 in Arabidopsis, and you knock out both of those genes, you don't get any root hair. So we identified direct targets of RHD6. And one of them is another basic helix continuous transcription factor called RSL4. So it's a distinct group, but they act downstream. And if you look at the transcription of RSL4 in an RHD6 mutant background, it's completely obliterated. So it's downstream, and the RHD6 protein directly binds to the RSL4 promoter, but I'm not going to talk about that today. If you knock out RSL4, you get this phenotype here. So here's our wild type, okay, and here's the RSL4 loss of function mutant. And there's two things to notice here, is that some of the root hairs are shorter here than they are here, and there are fewer. So there's a defect in initiation, and there's also a defect in elongation. Okay. So we were characterizing this phenotype, and in order to explain the phenotype, I need to talk through some basics of root hair biology that we discovered whilst we were doing this. So here's the root hair stem, and this is the elongation zone. The root hairs initiate here as bulges on the outer surface of the trichoplasts, and then root hair initiation is initially quite slow, so elongation in this axis, in this direction here, is quite slow. But then when they get to about 20 to 30 microns in length, then the rate increases and you get more rapid elongation here. And then growth stops. And so this process can take 8 to 12 hours depending on your growth conditions. And then there's a region here in which the root hairs are alive, but they're not growing. Okay, so these are middle-aged. They're not growing, but they're alive, so they're probably absorbing nutrients. And then they die. Okay, so these cells appear are all dead. So this is something I've been working on these things for 20 years, and I didn't know that these guys were all dead. So you grow them in the natural environment that is the Petri dish. They look perfectly happy. But as soon as you take the Petri dish off, these guys up here, the lid of the Petri dish off, these guys shrivel immediately, and these guys shrivel eventually. So there's nothing happening up here. So you've got this growth phase, and then this space where there's no growth taking place, but they're alive. Okay? Now, we figured that the RSL4 plant is required for root hair growth because the root hair is shorter than wild type. And so we predicted, well, if this is a positive regulator, then if we overexpressed RSL4, then it should make longer hairs, and it did. Okay, so here's what we see when we constitutively express RSL4. Okay, so initiation of root hair is around the same place as you see in wild type, and then this is the zone where they should be stopping, but they don't stop, they just continue to elongate, and they continue to elongate. So what's happening when we constitutively express RSL4 off the 35S promoter is that the root hairs stay in the constitutively growing phase, okay, and they only stop growing when they die. Okay, so this suggests to us that having RSL4 in a root hair meant that that root hair was going to grow. Okay, so it's like an on-off switch. If you've got RSL4, you grow. If you don't have it, you don't grow. And so that suggested that actually RSL4 protein should be present in these cells and then should disappear up here. Okay. And so that's more or less what we see. So here's a seedling. This is background down here. And we see the protein accumulate first in the elongation zone. And this is the zone where root hairs are initiated. And the protein is located in the nuclei. It's a transcription factor. It's where you expect to find it. Okay, up until around this region. So this is exactly where we expect it to disappear. Okay, so that looks okay. So here we've got some quantitative data to show you. So what we're doing here is this is the length of the root here. So this is where the expression starts. Okay, and then that's indicated here on this axis as time. So this is time 0 is when the expression just starts, and then this is, as you go up here, it's time since the initiation of expression. And so here we've got that, so time 1 hour here up through 12 hours. And what we're looking at here is GFP. So we see the fluorescence increase, decrease, and then disappear. Okay, and so at the same time as imaging GFP fluorescence, we were looking at the rate of, looking at elongation. So what we plotted here is root hair length. So root hairs are elongating here, so there's this initial slow phase, okay, and then there's this rapid phase of elongation, and then elongation stops right here. And this is exactly where RSL4 protein disappears. So the growth of these root hairs continues until the RSL4 protein disappears. So this suggested to us that actually the RSL4 protein really determines if the cell, the root hair, is going to stay in the growth state or a non-growth state. So then we have done some in situ hybridizations, and in situ hybridizations are a real pain in the ass to do in these plants. But we did them anyway, so you'll excuse the quality of this. But so what we're doing here is here's our wild type, and here's an RSL4 loss of function. So this gives you the background stain that you get when you do it in situ. Okay, but this really dark precipitate here indicates where the genes are, the mRNA is present. And then this is the site of root hair initiation. Okay, so what you're seeing is you're seeing message accumulate in a band just before and just after initiation. Okay, but I told you that the protein is present further up. Okay, so you've got a band of message, okay, and you've got a defined time in which the protein is present. So that suggests to us actually that what happens is you've got the message present down here, and a pulse of RSL4 protein is produced here. And the amount of that protein and the degradation rate of that protein determines the final length of root hairs. Okay? So you basically, early on, in this region of the root, you're deciding how long your root hair is going to be by making a certain amount of RSL4 protein. So what we decided to test this, so we predicted that all of our RSL4 protein would be made down here, that there would be no synthesis down in this region. Okay, so we did a FRAP experiment. So just to get you aligned here, so I'm using this schematic. Okay, so this is a pre-initiation. Okay, so this is down here. Here's initiation. You can just see a little knob building here, so that's there. And then we get this transition. So I told you this transition is this region where you're getting growth, but the rate of elongation is quite small. And then you've got these rapid growth phase here. Right? And so the experiment is to FRAP nuclei. So it's the photo-bleach nuclei at each of these stages. And if RSL4 protein is being made, then we should get recovery. So we should get green fluorescence returning in cells in which the RSL4 protein is being made. When we bleach cells in which there's no RSL4 protein, we should get no recovery. Okay, so here's what we see. So here's the pre-initiation, initiation, transition, rapid growth. Okay, and then across here we've got time. Okay, so this is a nucleus in this region, pre-bleached, then we bleach, the signal goes away, and as expected, the signal returns over a 15-minute period. Okay, if you look at the initiation zone, a nucleus in the initiation zone, GFP fluorescence, we bleach it, the fluorescence disappears, but it returns back pretty quick. Okay, then we go into the transition zone. So this is the zone in which the root hairs are elongated, but not really elongating very much. So we've got protein here, we frap, so we bleach, but we don't get any recovery, and we don't get any recovery up here. So this is suggesting to us, and so then this is qualified here. So these colours here match these colours. So for example, if we take the pre-initiation, which is the blue line, so fluorescence, we bleach it, we get very little fluorescence, but it recovers very quickly. Okay, we get the same, the initiation zone. But then if you look at the rapid growth into the transition zone, you don't get restoration. So this is consistent with the hypothesis that there's a pulse of RSL4 synthesis that's made right at the beginning of root hair development, and that root hair is elongated for as long as that protein is present. And as soon as that protein disappears, then root hair elongation stops. So we thought, okay, what sorts of factors are controlling the length of root hairs? And of course, there's, bringing colds to Newcastle when it comes to mind, talking about this here, of course, phosphate. Okay, so you grow plants on low phosphate, root hairs are considerably longer than they are if you grow them in adequate phosphate. So we decided to look and see how growing plants on low phosphate impacted this process here. Okay, so from this panel here, we've got some wild-type plants growing with lots of phosphate. Here's wild-type plants growing with not so much phosphate, and you can see that there's these hairs are much longer than these hairs. Okay, and it turns out that the levels of RSL4 expression are much higher in low phosphate than in high phosphate. So the low phosphate is increasing with transcription. Okay, so that's steady-state mRNA levels, but we really wanted to look at the protein levels. And so you've seen this form before. So this is fluorescence. So we're looking at GFP fluorescence here. Okay, and the grey bars, the pale grey bars, are the bars that these are plants growing in normal Arabidopsis medium. So you get an increased fluorescence, hairs that are elongating. Okay, and then after about 8 hours, the fluorescence disappears. But in the plants growing on low phosphate, I told you already that the steady-state levels of the messes are higher than in adequate phosphate. We overshoot here, so we get much higher levels of fluorescence here, so we're making more protein. Okay? And then that protein is much longer lived. So in these cases, it lives for almost 14 hours compared to 8 hours. So if our hypothesis is correct that having RSL4 protein present keeps the cell in the growth phase, this longevity, this increased longevity of the RSL4 protein could probably account for the increase in the root hairs. So we needed to check that this was actually reflecting reality. So we repeated the FRAP experiments. And so if you remember the previous FRAP experiments, we got recovery at pre-initiation, we got recovery at initiation. In the previous experiment, growing inadequate, we didn't get recovery here, but now we're seeing recovery. So what this is telling us is that the zone, this zone here, is making, our RSL4 protein is being synthesized in this transition zone where in adequate phosphate it wasn't. So we're making more RSL4 in the root hairs, and we're making it for a longer time than we did in adequate phosphate. And that results in the production of more RSL4, then is longer lived, and the root hairs grow longer. So our feeling is that we can account for, at least in part, the increased elongation of root hairs in low phosphate by the production of an increased pulse of RSL4 protein at this very early stage. So if RSL4 is controlling development, well, what sort of genes is it controlling? Well, it's controlling genes that are involved in cell growth. And I'm not going to go into any detail in this, but we've identified 34 direct targets. And a lot of these genes have been previously characterized. They're involved in the control of cell growth, and many of them are involved in tube growth, and here's a selection of them. So these guys up here, loss of function mutants, you knock them out and you get shorter root hairs than the wild type. And with some of these, if you overexpress them, you get longer root hairs than wild type. So our working model is that in this transition from here to here, we're activating suites of genes that are involved in growth, and RSL4 plays a key role in determining the growth state of the cell. If the RSL4 protein is on, it turns on, keeps the machinery turned on, and if RSL4 protein is absent, then the cells stop growing. So having done that, our next challenge, like I say, is to figure out what makes this structure give rise to a multicellular organ compared to this, and we haven't done that. So just as I wrap up, I just want to say that, well, I've talked about obscure model systems like Arcantia, we talked about synopsis, but actually a lot of this information does translate. So we've actually characterized these genes in the Poaceae. So this is work that we've done in Brachypodium, we've also done it in rice, and others have been doing it in wheat. So if you take RSL genes and overexpress them in Brachypodium, so see here's our wild type. These guys are bushier and hairier. You can see you get root hairs that are twice as long as the wild type controls. In addition, something that you don't see in Arabidopsis, if you overexpress these RSL genes in Brachypodium, you transform every cell in the epidermis into a root hair. And then that's indicated here. So this is a proportion of cells that are root hairs in grey and non-root hair cells in white. And there are very few of these. This white bar up here is really small. So this indicates that you could begin to use these genes to modulate root hair characteristics in the cereals. And we've started to do this, and we've made some constructs in which we express these genes only in the trichoblast cells. So we're going to get long root hairs, but we don't get ectopic root hairs. We don't get these supernumerary root hairs. And it's very early days yet, but when we do these experiments in the greenhouse, we begin to see slight enhancement of growth in certain conditions. And we're collaborating with Peter Ryan at CSIRO to do this properly. He's going to do it properly. So up here, we've got just in these boxes here. So this is above ground biomass. These are the plants that are expressing RSL genes in the root hairs alone. So the only difference between these guys and wild type is they produce the same number of root hairs, but those root hairs are much longer. And we're seeing in a variety of conditions slight increase in biomass if we make these longer hairs by overexpressing these genes. And we're seeing slight increases in total brain weight. Okay, so this is all Brachypodium. It's done in the greenhouse, so it's not really Brachypodium. It's not done in proper soils, but Peter is doing these in soils. And we've also done the same in rice, and we're seeing similar changes in rice. And we're doing some field trials in Korea to see does this actually make any difference in the field. So it's time to wake up for those of you who've gone to sleep. And as I said at the beginning, the aim was to figure out what was happening back here. And what I can say is that back in the common ancestor of the land plants around 470 million years ago, they did utilize the RSL genes to promote the development of these rhizoidal cells. And we think that not only were they controlling the formation of rhizoidal cells, but they were controlling the development of many cell types that develop, or many structures that develop from a single epidermal cell that broke the plane of the epidermis as it developed. And then I went on to tell you that we can begin to use these to modulate root hair characteristics in a variety of species. And I'd like to thank you very much again.
question is there any evidence that these kind of basic genes are also involved in the after elongation, like in the animal system or
Okay, so they're not involved in Arabidopsis trichome development, and in Arabidopsis they're not expressed in the lead. In the grasses, there's two members of the class 1 RSLs that are expressed in the shoot system. So now we're looking at the Brachypodium and rice plants to see are there any developmental processes that are defective there. So that's work that's ongoing. We haven't looked at any of the other bryophytes apart from these guys here. But we have the material now to begin to address this in the vascular plants. The pathway that controls trichome development, or at least trichome spacing in Arabidopsis, is very derived. So it only evolved very late. So it's not general for tracheophytes. So it'll be something that evolved in the ancestors of the Brassicales. So it's a very specific example. So I'm not too concerned about that. These basic helix proteins operate in animals. So this group of basic helix transcription factors are plant specific, so they don't really look like the basic helix transcription factors in animals. But there is a basic helix transcription factor in animals that is a master regulator of neuronal growth, not in the same way that ours is, but it has a very similar role in controlling ultimate cell size. So I think it's an example of convergence, but there's only one example that I know that shows any similarity.
RSL in algae has any function at all?
Pardon?
Does RSL have any function in algae?
Oh, an algae, sorry. That's a lovely question. So we found if you look at the basic helix domain of the RSL genes, there's a very specific basic helix domain, and next to it there's a domain that we call the RSL domain. So it's a short run of amino acids that we don't see in any other basic helix proteins. If you now look at the algae, in the stratified algae, we've got proteins that look just like our basic helix proteins, but they don't have the RSL domain. So the difference between Cara and Coleochaete RSL proteins is that basic helix is the same, but they don't have this short run of amino acids. And if we now take the Cara and Coleochaete protein and overexpress it in Arabidopsis and in Mercantia, we do not complement the mutant phenotypes in loss of function mutants, so we think that they're functionally different. Also in Cara, which has structure, if we look at expression, the RSL relative in Cara is not expressed in the Cara rhizoid. And it's actually expressed around the reproductive structures. So this now begins to make sense insofar as if we consider that the RSL proteins are not rhizoid or root hair specifics, but there's something more fundamental to do with cellular outgrowth that might explain that. But there is something distinctively different about those proteins in the algae. We don't see them in the chlorophyte algae, so we found them in the stratifieds.
I was wondering about the RSL overexpressed phenotype, whether it's specifically related to increase in RSL for increase in root hair, increases the specific phosphate uptake, or it's also associated with other micro and macronutrients.
So we haven't done nutrient uptake experiments yet. So RSL. So if you overexpress RSD6, you get longer root hairs, and you get increased steady-state levels of RSL4. So I think increased RSD6 increases RSL4, increases root hair length. What we haven't done is measure the impact on phosphate uptake. And but theoretical models would suggest that in low phosphate soils, you may see an impact there, but we haven't done that yet. Pete might talk to you. Maria.
Does phosphate influence the rhizome growth in Marcantia? Can you use that system to get to phosphate sensing?
Yeah, so I think the answer is yes. So if you grow these plants on low phosphate, you get they look like more rhizoids, longer rhizoids, but these are indeterminate structures, so it's a bit hard to show. But you do, so you increase them. We've got a transcriptome of the rhizoid, so we're seeing all the phosphate transporters in there. And also we're seeing expression actually of genes involved in nitrogen assimilation actually. So we think these really are rooty things. And what I'm particularly carried away about at the moment is, I don't know if you noticed, but that screening was done with 300,000 tDNA lines. You're not going to do that today or tomorrow in a Arabidopsis or in a Cayuga. And also, we've got UV and EMS mutagenesis working, which makes it much easier. So for me, we're getting geared up. We want to do a few selection screens. So we've done some screens. So we've done one screen where we had a selection. It doesn't matter what we're selected for. So basically, everything dies except what you want. So we were able to screen 2 by 10 to the 7 individuals. We got 23 alleles in one particular gene, and we sequenced 6 individuals. And by just sequencing 6, so I had a feeling what the target was, but I said to the student, I said, look, just see, can you find out what it is by just comparing SNPs? And so he said, yeah, he did, and he found the target. And he could have done it with 5 alleles. We had 6 or 7 alleles. So I think this is going to be a really great system if you've got a really nice selection. So, arsenate comes to mind, and there are other things. So I think if there's a nice screen that's not too leaky, and it's haploid, and you can just see, you can still do bulk segregant analysis if you want to. So I think this could be quite useful. Also, one of the things that we see is in some families, limited redundancy. Fewer numbers of genes suggestive of redundancy. So that's good on the one hand, insofar as you may find the mutants. Okay, you might say, well, it's bad because you knock out something, then you get lethality. But of course, if you use EMS and UV, then you've got a chance to find quite more. So that was our motivation for doing that. So I think the genetics in this system is really, really powerful. And in a way, doing this stuff is this is the dull stuff. Now I think we can start to do really elegant screens. So if you've got ideas for really neat screens or selections, we can help. Let me know.
So in the frag assay, you have shown these, first bullet shown these nuclei. And I think the nuclei has 2 different structures. One is the in transition, especially John. One is the elongated in the previous slide, and the later slide it's getting a more rounder. So do you have any idea why?
So then in the meristem, the nuclei are perfectly spheres, very big spheres, huge nucleoli, and then as they go through the elongation zone, the nucleoli shrink, and then the nuclear volume decreases, but they're still spherical. And then just after around the time of initiation, the nucleus begins to elongate, and then when it moves into the hair, it's completely elongated. So you see that pretty reproducibly. What we don't see is we don't see any statistically significant difference in that shape change in the low phosphate plants. Our interpretation is that it just before it initiates, it begins to elongate in a spindle shape, and that's what we determined is, but we haven't found a reproducible effect in low phosphate, but it's an interesting question.
Well, if no more questions, let's thank Professor Dolan for reaching out.